Welcome to Solutions OP Clinical Trials Podcast. My name is Olga and I'm the Managing Director of Solutions OP. We are a small team of experienced professionals working on clinical trials in the United Kingdom. I'll use our podcast to discuss hot topics in clinical trials and provide analysis of the latest trends and challenges in pharma and medical device industries, not just in the UK but globally. Thank you for listening. Welcome to today's episode of the podcast. Today I'll be talking about cell and gene therapies and also about their potential as new treatments and the drug development challenges with these products. These relatively new treatment options are very popular and they attract a lot of investment at the moment. It's a field where we have some success stories, but we also have some failures. So it's very interesting, I think, to learn a little bit more on what's going on at the moment in the field and the latest trends and also the biggest challenges that companies face. I will try to keep it as less terminology as possible, and I do that on purpose because I want to make sure that even people who don't have the medical background or who don't work in the field, they can still feel comfortable and understand what I'm talking about. So let's start with the current situation with clinical research when it comes to cell and gene therapies. So the number of clinical trials is growing with 337 clinical trials in Europe in 2025. For the same period in the US, there are between 700 and 1,000 cell and gene therapy clinical trials which were ongoing. So as you can see, there's a great interest in the area and there are plenty of clinical research going on. So according to the UK statistics, many of the clinical trials are gene therapies, and there are almost 50-50 in vivo and ex vivo. In a moment, I'll explain a little bit more about in vivo and ex vivo methods and all that. So the next big group of clinical trials are some somatic cell therapy therapies. And also the smallest group is the tissue-engineered therapies. In terms of indications, the main indication is oncology. And I think because there are some success stories with oncology treatments, it's normal to have more oncology cell and gene therapies in development. And also it's a field where we need some breakthrough. But there are also some other fields which are gaining momentum at the moment, like hematology, autoimmune diseases, and ophthalmology. So ophthalmology, these are the eye diseases. For those of you who don't know what that fancy word means. So what types of cell and gene therapies are out there? That's the next thing I want to talk a little bit. So the gene therapies are divided into in vivo, when the gene manipulation happens in the body of the patient, and ex vivo, that's when the manipulation is outside of the body of the patient. The chimeric antigen receptor T cell therapy is also known as CAR T cell therapy. Is used to reprogram the patient's immune system and to help fight some types of blood cancer. And there's some approved therapies on the market. So the CAR T cell therapy is divided into allogenic when it uses donor cells, and autologous when it uses patient's own cells. If I remember correctly, the first successful story was back in 2015 with autologous cells, and it was a little girl that had a blood type of cancer which was treated. So however, last year some pharma companies started using in vivo CAR T cell therapy technology, which is basically manipulating patient cells in the body to produce CAR T cells. So I'll talk a little bit about that later on in the podcast if you're interested to learn a bit more. So while the autologous therapies are available on the market, the allogenic option is still in development. So the allogenic is when it uses donor cells. The development of allogenic options is important, particularly for mass production, and also because that has the potential to reduce the overall cost of CAR T cell therapies. So the current available options on the market are autologous CAR T cell therapies. They use the blood sample or the blood cells of the patient, which are programmed to attack the cancer cells. And these therapies are very expensive, as you learn if you continue listening. I'll tell you more about the costs in a bit. So what are the main differences between the autologous and allogenic cell therapies? So the autologous cell therapies are using, as I said, patients' own cells, and they're normally slower in terms of action. But the response from these treatments is longer in comparison to allogenic cell therapies, which use donor cells. And another thing about the allogenic cell therapies is they act faster, but their response is short-lived at the moment from what's been observed in early research. So just to summarize, so the autologous ones, which use the patient's own cells, they take a long time to prepare, because the researchers have to isolate good cells from the patient, gene modify them, and then return them to the patient, and it's a slower response for that type of therapy, but the response is long lasting, and it has a high efficacy because it basically activates the immune system of the patient. And the most positive part is that there is no immune rejection. So because these are cells which are already derived from the patient, there is no rejection. For allogenic therapy, which use donor cells, it's different. So they are available immediately off the shelf. So the idea is that you use donor cells, they are modified, and they could be used for a bigger proportion of patients, so it's not just for a specific patient. The response rate is much faster, and also, unfortunately, their effect is shorter because they're cleared a lot quicker by the immune system. And they have initially good response, which is not that durable, so it's not a lengthy response, and potentially might need additional doses and so on. So the other complication here is the immune rejection. Because you're using donor cells, there is a chance that these cells could be rejected by the patient. So one of the potential benefits of the allogenic cell therapies is that it's off the shelf, as I mentioned, and also they allow to improve the safety profile and the potency of these therapies, and make them available for more patients, more than one patient, which potentially could reduce the overall cost. Now, that's a tricky one, because if you have a shorter response and you have to give additional doses to patients, it might be that they will not be that cost effective as they sound, unless they improve to such extent that they have a longer response. So the other topic I wanted to touch is what are the new emerging trends in cell therapy. And here I'll review some therapies which are currently in development. So chimeric endocytizing membrane antigen cell therapy is also known as cema-cel. And this is a new method eliminating diseased cells by removing a protein from their surface. So this new approach is used in some cancer types and also autoimmune diseases and infectious diseases. So what is the difference between the existing CAR-T and the cell? So the CAR-T basically, the cells are activated and they kill the target, while the cell works in a different way. It removes an antigen, a protein which is on the surface of the cells. As a result of the removing of this antigen, then the cell is able to kind of use endocytosis and modulation to basically the therapy gets in and allows the cell to kind of consume itself in a way, if you want to put it that way, in very very unscientific way to say it. So it works in a different way. So basically it affects the cell, while in the case with CAR-T therapy we have immune activation, and the cells are targeted and then killed. So for CAR-T therapy, current use is mainly for cancer, while for the cell, they're hoping that this mechanism could work for various diseases, like, as I mentioned, autoimmune diseases and infectious diseases as well. So the CAR-T is obviously approved already on the market, which is good. And the cell is still early research, so we don't know how it will develop, if it will be successful or not. We will learn in the future how this field develops. Another type of cell therapy which is in development is anito-cel, which is autologous CAR-T cell therapy. It uses patients' own cells, and it's currently in development for multiple myeloma. The T cells are collected from the patient, and then they're modified in the lab to recognize and destroy a protein, B cell maturation antigen, which is found on the surface of the myeloma cells. The cells are then infused back into the patient, and this new method improves the binding to the cancer cells and is expected to have a better safety profile. So far it has shown very high response rates, and many patients have complete remission, which is fantastic. It's submitted for approval with the US FDA. We'll see what they'll decide and what's going to happen. Hopefully there will be some news on this in 2026. So we'll see how this will develop. Some other types of cell therapy which are currently in development are Lesmi cell and ETI cell CAR-T cell therapies. They're all allogenic therapies using donor cells. Both target B cells protein, which is responsible for preventing the overactivation of the immune system. And this is an important biomarker for leukemia and lymphoma. So these both therapies are currently in development, still early stage, for leukemia and lymphoma. So we'll see what happens and what will happen with them. There are some other therapies as well which are at the moment in development. So that there's a lot going on in the field. Now the next important topic that I want to touch is what are the challenges with cell therapies. And this is a very important one, because I think we need to understand the complications and but probably will give us a bit of an idea of potential future of cell therapies as well. So the first big problem is the quality of patient cells. So the biggest challenge for allogenic CAR-T cell therapies using patients' own cells is that often cancer patients don't have good quality immune cells that could be used to prepare the treatments. So one of the reasons is that because patients had previous therapies and chemotherapy, it normally affects the immune system, and it also cancer itself affects the immune system. So inducing immune response to fight the cancer cells could be a significant challenge when patients have exhausted immune system. And just finding the good cells that you can use to activate and then to activate the immune system of the patient could be very challenging. All these CAR T cell therapies currently are approved for patients with more advanced cancer, which makes it more complicated. So they're not like first line treatment, they're for patients with more advanced cancer. But again, when it's more advanced, then the immune system of the patient is in a worse state than if they're treated after initial diagnosis. Another important point here is the way cancer progress. The way different cancers progress is different. Sometimes it's not necessary to start treatment immediately, which makes allogenic CAR T cell therapies appropriate. In some cases, the physicians prefer to have an off-the-shelf option, and autologous therapies will be preferred. The allogenic option will be best for fast-progressing cancers, because if you have ready off-the-shelf therapy that you can give to the patient straight away, and you know that patient has a fast-progressing cancer, you have a better chance for success to do something. You can't keep that patient for a long time, because then they'll further progress, and the chances of the therapy working reduce as the patient's health deteriorates. So that's important to remember. Another important complication is the costs. Actually, the cost is quite significant. So for autologous therapies, which are basically derived from the patient, the cost, which I'll tell you, is based on therapies which are already on the market. It could reach $500,000 and it could be even beyond that. That includes not only the treatment cost, but also the whole stay in the hospital and the whole associated treatment. So there is an estimate that the total treatment cost could be over 1 million, and that's for one patient. So as you can imagine, it's really expensive. The manufacturing is patient specific, so that's why it's so expensive, because you have to prepare it for the specific patient. You have to harvest the immune cells, modify them, infuse them back in. All that takes time and work, and as we said already, some patients don't have very good quality immune cells anyway. And availability takes weeks to prepare, so it's not something that is very quick to do. Usually it's one-time treatment. That's the other thing, and probably that affects the cost as well. If it's a one-time treatment, it tends to be more expensive. For allogenic therapy, when you're using donor cells, now first to say that these are still in development, so there is no allogenic therapy on the market yet. So the cost is projected cost, so we don't know the actual cost if these therapies reach the market. But the projected cost is up to $150,000 US. And overall treatment is expected to be a lot lower than 1 million. So hopefully that will be less. Manufacturing, it will be a mass production if these therapies turn out to be good. So that's another positive thing. If you can have mass production, they should be available immediately, so there's no need to wait for them. But the dosing might require more doses. So as I said, if you need more doses, then would that be that cost effective? We don't know yet, because we don't know any any product approved on the market, but I guess we will learn. Another important challenge when it comes to cell and gene therapies is the rejection of donor cells is a big challenge for allogenic cell therapies. So one solution to this problem is to remove protein on the surface of the cells, which is used by all cells to be marked for destruction. However, this will affect the natural mechanism by which other cells of the immune system, called the natural killers, how they act, because they will destroy all cells that lack this protein. So what happens is, if I remove this protein, it will allow me to get the cells into the body of the patient without a rejection, but that will activate other cells of the immune system to attack them and destroy them. So it's a very tricky situation and very complex. And while you think it's inconvenience, you have to remember that that's the whole idea of your immune system. It's there to protect you. So I think the immune system is working right, but for the purpose of cell therapy, it could be a nightmare. So some companies have tried to create "don't eat me" signal to prevent the natural killer cells from attacking the modified cell therapy cells. But there are different types of natural killers, so not all of them recognize this signal. So that's another complication. It's just you have so diverse cells of the immune system which are designed to be able to detect any problems and attack these problems, which is a good thing, as I said, but it's a bad thing if you're working on cell therapy. It's also a bad thing if you happen to have autoimmune disease, but that's a completely different story. Another challenge I would like to discuss is the in vivo CAR cell therapy, which has its own risks. So in the in vivo CAR T cell therapy, the T cells are modified in the body of the patient. So this means that hundreds of trillions of DNA are injected into the patient. One challenge here is that the viral vector, which is used to introduce this new DNA into the cells, could be at risk of integrating into the genome of the patient. And this could affect many inactive oncogenes and potentially risk inactivating them. And this has been observed with patients already. So what happens is we have in our genome oncogenes which are there, but they're not active and they don't cause cancer, basically. But if we have a vector which integrates into who knows where in the genome, then that could affect some oncogenes which are inactive and potentially activate them. So in clinical trials, they have observed such cases where patients are treated for one cancer and they've detected that the vector has integrated into the genome, and there are some different types of cancers that have developed as a result of this integration because some oncogenes have been activated. Because it's early stage research, it's still not known to what extent, how big is this problem. So we don't know yet, is that like 50% of the patients who experience, or is it going to be 1% of the patients who experience? So it's still early stage, so we don't actually know, we don't have any reliable statistics to say what's the risk. But it's shown that there is a risk, and we know there is such risk. That's the important part. So another problem is, in order to import that vector and the new DNA into the body of the patient, you need to use lipid nanoparticles, which use like a delivery system. So they're like a membrane of a cell, and they wrap everything around, and that's how they're imported into the body of the patient. However, these lipid nanoparticles, they tend to accumulate in the liver, and this could increase the risk of liver toxicity. So that's another problem which is related to delivery system, how things are delivered into the patient. So as you can see, in conclusion, what I can say is that cell and gene therapies, they look very promising, but we are not there yet in terms of actually massively use them for different diseases and different patients. There's still a lot of unknowns. It's still early stage development, and there are potential risks, as we discussed. And the cost of the therapies at the moment is quite significant, and that could be a particular big challenge, regardless if you have public healthcare system or you pay for yourself. 1 million for treatment is something unaffordable for many people. There is a potential in cell and gene therapies, but as I said, there's still a lot of unknowns, and there's still a lot of more work that needs to be done before we have clarity how this will work. Maybe in the future there will be some new technologies which are developed, and there will be a different way that we will be able to manipulate cells and attack cancer in a different way. But at the moment, the existing methods are, let's say, not ideal. But of course, it's a field which is developing fast. So I think we'll see a lot of more exciting news in cell and gene therapies in the future. Thank you for listening. I hope that was interesting.